This video is about the Bohr shift, also known as the Bohr effect. So it's something about the blood, and this was discovered by this gentleman over here. His name is Christian Bohr in 1904, that he found out that in the presence of carbon dioxide, the oxygen affinity of hemoglobin decreases. So it's basically when you have a lot of carbon dioxide present in a tissue, that carbon dioxide causes oxygen to release, it causes hemoglobin, to release oxygen. So if you have oxyhemoglobin in the presence of carbon dioxide, that oxyhemoglobin dissociates and releases its oxygen. Alright, so let's find out exactly how this works. So there's this mechanism here. So first let's think about what happens with carbon dioxide. So you have the blood plasma, right? And you have respiring tissue and the release carbon dioxide. So you have red blood cells that's there in blood. Now within red blood cell, you have a special enzyme, and that you have to, store it in your memory, right now, long-term memory. This enzyme is called carbonic anhydrase. Okay, so what this enzyme can do, it can actually speed up, the combination or the reaction of carbon dioxide and water to give us carbonic acid. Okay, it's a reversible reaction. So once carbon dioxide gets into the red blood cell, this enzyme helps this to happen. So carbon dioxide plus oxygen, you get carbonic acid. Now then, that acid, what it does, it basically dissociates and it releases H plus ions, hydrogen ion and hydrogen carbonate ion. So basically what you've seen is that the carbon dioxide has ultimately been converted into the hydrogen carbonate ion. And guess what, boys and girls? That hydrogen carbonate ion is basically the major form in which carbon dioxide is carried in blood. In fact, 85%, what was that? 85% of carbon dioxide, carried in blood is carried in the form saying hydrogen carbonate ion. So 85%, and that's possible thanks to carbonic anhydrase. Now, what you can notice here is that during that process, H plus ions are released. Now, if you basically refer to your basic chemistry, what happens when you have a lot of H plus? What happens? The pH drop, right? You get a low pH. That means the solution becomes acidic. Now, that's not a great thing because we're talking about this reaction happening in the blood. So therefore, your blood will tend to become more acidic every time carbon dioxide, right, is being converted into hydrogen carbonate ion. So therefore, to get around this problem, guess what? There is, obviously, nature has already thought about this, and basically what will happen is the carbon dioxide, the H plus will combine with hemoglobin. So the molecule hemoglobin combines with that H plus to give us hemoglobinic acid. So here, what you'll notice here is because, it's basically when the hemoglobin combines with H plus, forming hemoglobinic acid. That means if that hemoglobin was combined with oxygen, that H plus will basically cause the oxyhemoglobin to release its oxygen. So consequently, once we go, down back to the first step, what we can conclude is we can say that the carbon dioxide basically triggered or caused the oxyhemoglobin to dissociate to release oxygen. So basically it means if your blood is oxygenated, you need carbon dioxide for that hemoglobin to release its oxygen. That oxyhemoglobin to release its oxygen. It actually speeds up the process. It enables this to happen, all because of that hydrogen ion that is formed. Alright, so that's basically what you're seeing here is the Bohr's effect. This mechanism here. Carbon dioxide gives you carbonic acid dissociates, that H plus combines with hemoglobin, the protein, causes the hemoglobin, again, a distortion, and that causes, if ever the hemoglobin is combined with oxygen, it causes it to release oxygen. So that particular mechanism here has it's significant in two ways. Let's see. So number one, that means hemoglobin is acting as a buffer. Okay, a buffer is basically a solution that controls the pH. So somehow blood itself does not become acidic thanks to hemoglobin, because the hemoglobin is gonna combine with that H plus that is formed in order to form hemoglobinic acid. So that means hemoglobin is acting as a buffer. The other significance of this particular mechanism here is basically the Bohr's effect. So it means where you have a high partial pressure of carbon dioxide, so where you have a lot of carbon dioxide, what it means is if you have a lot of carbon dioxide, it causes hemoglobin to release oxygen. So it's very logical, and it makes perfect sense. Therefore, we can say in respiring tissue, wherever there's a lot of carbon dioxide with a high partial pressure, it means the oxyhemoglobin that's there will release and dissociate and release oxygen. So therefore, supplying oxygen wherever it is in demand. So you can see this mechanism somehow helps in regulating how much oxygen will be released by hemoglobin. Okay, so wherever the oxygen is needed more, okay, more oxygen will be released by oxyhemoglobin, because in this area where we need more oxygen for respiration, there's obviously more carbon dioxide. Okay, so hopefully that makes sense. Let's see. All right, now that we've understood the effect of carbon dioxide on the release of oxygen, let's find out the effect of this on the dissociation curve. So what you can see is basically, let's see the effect of concentration on hemoglobin saturation. So when you have a low concentration of carbon dioxide, you can see the curve, okay, is like this. Okay, so at 14 kilopascal, 95 saturation, at 4 kilopascal, you have a slightly higher saturation. But then in the presence of carbon dioxide, with a higher partial pressure of carbon dioxide, what happens? It causes this dissociation curve to shift to the right. Okay, so it is lower than normal to the right. So if we compare here, just look, for instance, at 4 kilopascal, with a higher concentration of carbon dioxide, the saturation is lower. Look at this. You can immediately see. Likewise, at any point on the curve, you can see that compared to the normal curve, okay, the curve with the higher partial pressure of carbon dioxide, okay, will always cause the hemoglobin to have a lower affinity for oxygen. So basically, in the presence of carbon dioxide, hemoglobin tends to combine less with oxygen. So also, what this means, what's the significance of this? This is what it means. Look at this. So normally then it means that when you're in the lungs, where the partial pressure of oxygen is high and the partial pressure of carbon dioxide is low, right? So low and partial pressure of oxygen is high, then in this case the affinity for hemoglobin for oxygen is very high, so saturation will be very high. However, when you are in active tissue, what it means, the condition are you have a high partial pressure of carbon dioxide and you have a lower partial pressure of oxygen. That means the curve, the dissociation curve shifts, it like gets lowered to the right, and that means then in active tissue, at a low partial pressure for the same, you can see here, instead, for instance, if let's take 2 kilopascal, with a normal curve, normally the saturation should have been like 23%, but then because of the Bohr's effect, okay, because of the carbon dioxide, the saturation is less. That consequently means what? It means that in the presence of carbon dioxide, okay, hemoglobin tends to release its oxygen. So that is very convenient, okay? So that's basically the significance of the Bohr's effect, okay? It basically means that carbon dioxide will be, oxygen is released when there's a lot of carbon dioxide, and when there's less carbon dioxide and there's more oxygen, then hemoglobin will tend to take up oxygen. So this ensures that the right amount of oxygen is released, okay, within the body, okay, at the right site. For instance, let's look at an example of basically the transport of oxygen and carbon dioxide between the lungs and respiring tissue. So look here. Here we are in the lungs, right? In the alveolus. So in the alveolus, you have a high concentration of oxygen, high partial pressure of oxygen, and low partial pressure of carbon dioxide. Okay? So it means then, with that high partial pressure of oxygen, it means oxygen will tend to combine with hemoglobin. Now, if you remind yourself about this whole process where how hemoglobinic acid is formed. So that hemoglobinic acid is there when blood reaches the lung. It is obviously deoxygenated blood, right? It is deoxygenated. So basically that deoxygenated blood means it has a low saturation of oxygen, and obviously it carries more carbon dioxide in the form of hydrogen carbonate ion. And also, basically, you know that the hemoglobin is in the form of hemoglobinic acid. So the oxygen combines with the hemoglobinic acid. That causes the hemoglobin to combine with the oxygen and release the H plus ion, right? That H plus ion will combine with the hydrogen carbonate ion that's there. So hydrogen carbonate ion will diffuse from the plasma into the red blood cell. And guess what? We have our enzyme, the carbonic anhydrase. So it gets hydrogen carbonate ion to combine with H plus, and this gives you basically water and carbon dioxide. So basically then the carbon dioxide will diffuse out of the red blood cell and ultimately pass into the lungs. So that's how basically oxygen is taken up and carbon dioxide is released, from the blood into the air, into the alveolus. So likewise, let's look at the last example, the reverse of this process. Sorry, let me close this here. Look here, the reverse happens. So basically now, let's say you are in the respiring tissue. In respiring tissue, right, respiration happening, a lot of carbon dioxide is produced, right? So this carbon dioxide here, so in this condition, you have high partial pressure of carbon dioxide, you have low partial pressure of oxygen. So that means, if you look on the curve, we are like here. So at this point, at a low partial pressure of oxygen, and on top of this you have a high partial pressure of carbon dioxide, Bohr's effect, it causes the hemoglobin to release its oxygen. So basically look at this, carbon dioxide gets into the red blood cell, combines with water, in the presence of carbonic anhydrase, you get hydrogen carbonate ion and H+. What will that H+ do? It goes combine with hemoglobin, like there's a oxyhemoglobin, right? It combines with that hemoglobin, it causes like a distortion, thereby causes oxyhemoglobin to dissociate and release oxygen, and you have hemoglobinic acid formed. So the oxygen then leaves the red blood cell and it feeds to the respiring tissue. Alright, so that's basically how oxygen is taken up and released from the red blood cell.